Namaste. In the previous class, we're discussing about nature of the seed treatment. In the case of the seed treatment, there are the various techniques. Some are the mechanical scarification method, some are the chemical scarification method, and vernalization of the seed treatment with the biofertilizer, seed treatment with the growth hormones, seed treatment with the biofertilizers, like that, and we're having the biopriming technique. So with the help of this technique, right, so we can able to minimize the contamination and enhance the shelf life of the seed can be possible. So apart from that, we're having the 10 marks question we called as isolation distance. So here in the C technology, we're having maximum 10 marks we can expect in the UPSC. So in the case of the isolation distance, the minimum distance will be maintained between the one field to the another field, right? So minimum distance is maintained between the one plot to the another plot to avoid the contamination, to avoid the mechanical contamination and to improve the homogeneity in the seed lot is called as isolation distance. So isolation distance is the one of the method in the genetic principle, is the one of the method in the genetic principle for the quality seed production. Depends on the nature of the crop. Nature of the crop pollination, we're having the some are the self pollinated, some are the cross pollinated. In the case of the self pollinated, right, the minimum 3-meter distance we need to maintain between the one field to the another field, then only we can able to minimize the contamination in the seed lot. But in the case of the cross pollinated, we require the more isolation distance between the one field to the another field. There we require minimum 600 meters. 600 the meter, the minimum distance we need to maintain between the one variety to the another variety. In that cases, as much as possible we can able to minimize the contamination in the seed lot. So it is a one of the method in the case of the genetic principle, in the case of the quality seed production. So in the UPSC, for example, in the isolation distance, right, so we can expect what you understand by the isolation distance. So in that cases, the minimum distance is maintained between the one variety plot to the another variety plot. Then we're having the types of the isolation distance. So within that we're having the three type of the isolation distance can be possible. Then we're having the some example, right, with the help of the minimum distance. So what kind of the contamination we are minimizing in the different crops. Then apart from that, we're having the significance of the isolation distance. So in the case of the isolation distance, right, as much as possible with the help of this, so we can able to minimize the contamination and maximize the physical purity and genetic purity in the seed lot can be possible. Then isolation distance for the major crops. Major crops matlab we are having the some are the self pollinated, some are the cross pollinated, some are the upon cross pollinated crops. Then we are having the factors affecting the isolation, the distance. Then we are having the circumstances for the increase or decreasing the isolation distance in the given condition. Then conclusions here, right? See, whenever if they ask for any questions on the isolation distance, because in the previous UPSC paper, as of now, they asked for 2 times. They asked for the 2 times for the 10 marks question, right? So in the 2 pages, right, so these are the elements we need to take for the given presentations here, right? So here, so in the isolation distance, as we said that it means, right, the minimum distance, right, it is a more of the minimum distance is maintained between the two varieties. Between the two varieties, there is a minimum distance we are maintaining between to avoid the mechanical mixture, and to improve the homogeneity in the given seed lot. Homogeneity matlab kind of the physical purity and genetic purity can be enhanced in the seed lot with the help of the minimum distance between the one variety to the another variety is called as isolation distance. In other words, right, this is one of the aspect of the genetic principle. I'm repeating once again, right, this is one of the method or aspect in the genetic principle, in which there is the minimum separation matlab the minimum distance is maintained between the one field to the another field for the quality seed production in the seed technology, right? So with the help of the isolation distance, right, as much as possible we can able to minimize the contamination, right, pollen contamination and the disease contamination can be minimizable in the case of the seed production. See, if they ask for what you understand by the isolation distance matlab, then you have to write this much in the given pages it is. So here, so the distance will be maintained between the two seed crops, and different seed production of the same plant variety and in the location to prevent the deterioration of the genetic purity. As much as possible, the minimum distance is maintained between the two variety to minimize the deterioration in the genetic purity in the given seed lot is called as isolation distance. So it is different, right? There is a different isolation distance we have for the different crops, right, based on the nature of the pollination. Based on the nature of the pollination, we are having the three type of the crops can be possible. Some are the self pollinated, some are the cross pollinated, some are the upon cross pollinated crops can be possible. According to the nature of the pollination, right? There is a different kind of the isolation distance here, right? For example, we are having the rice crop. Rice is a more of the self pollinated, so there we require minimum 3-meter distance. We need to maintain between the one variety to the another variety of the rice crop. And in the case of the wheat also 3 meter is sufficient. And we are having the cross pollinated based example bajra. In the case of the bajra, minimum 1000 meter we need to maintain between the one plant to the another plant, right? So like that, the minimum distance we need to maintain between the one field to the another field it is. So here in the given slide we are having some example and case study. Whenever if you follow the minimum distance between the one field to the another field, right? So with the help of the isolation distance, one can able to minimize the contamination, in the seed production. That is, so we can able to minimize we can able to make the quality seed production in the case of the wheat crop from the loose mud. As much as possible, contamination from the loose mud can be minimizable in the case of the quality seed production of the wheat crop. Then contamination from the grain smut in the sorghum can be minimizable. Then contamination from the alternaria blight in the case of the mustard seed production. Then early blight and leaf spot contamination in the tomato can be minimizable. Then brown rot and root knot nematode in the case of the potato crop can be minimizable. Then yellow vein mosaic is one of the viral disease in the bendi can be minimizable. The little leaf, right? In the case of the brinjal, little leaf is one of the mycoplasma disease, So in these cases, as much as possible, all the kind of the contamination in the quality seed production can be minimizable. So these are the some case studies here, right? Whenever if you maintain the minimum distance between the one field to the another field, so contamination in the given seed lot can be minimizable. So that homogeneity, matlab physical purity and genetic purity in the seed lot can be enhanceabilities. So here within the isolation distance we are having the three type of the isolation distance can be possible. One is the distant isolation. Distance matlab minimum distance we need to maintain between the one field to the another field. We said some are the 3 meters, some are the 500 meters, 600 meters. So minimum distance will be maintaining between the one crop to the another crop based on the nature of the pollination crop. Based on the nature of the pollination crops, the minimum distance will be maintained. Then we are having the time isolation. Time isolation and the staggered sowing time, right? So for example, you are planting one variety in the month of February. Another variety will be planted in the month of like April. So the minimum staggered differential planting time is required. In that cases, we can able to minimize the contamination or the time of the flowering. Then we are having the barrier isolation. We are having the barrier isolation. In the barrier isolation, right? So for example, you are taking the quality seed in the center of the farm area, and across the boundary of the farm gate, we'll be planting some dense crop. Like we're having example maize. The maize will be planted as a barrier crop across the boundary of the farm area, and within the farm area you will be taking some quality seed, like you are having some vegetable or whatever it is, like tomato, potato will be taken within the piece of the land. Across the boundary as a dense barrier crop will be planted as a maize. So that nature we called as the barrier isolation. In the case of the barrier isolation also, we can able to minimize the pollen contamination. So we are having the one is the staggered isolation, another is the minimum distance, another is the barrier isolation. These are the three type of the isolation distance we have here. So here, whenever if you follow the isolation distance, right? So these are the some significance can be possible. One is that it helps in preventing the cross pollination from the one variety to the another variety. Then maintaining the physical purity. Physical purity means physical composition of the seed lot, and can able to enhance the genetic purity of the crop. Right? Can able to enhance the genetic purity of the crop. Then maintain the true to type of the character. Maintain the true to type of the character in the given seed lot can be possible. Then no contamination from the uptype of the plants. Uptype of the plants means we are having the mutant plants. We are having the example we call as mutant plants. Mutant plants means some are the taller varieties, some are the dark varieties. Like whatever the mutant plants will be there, they're called as uptype. Even include we are having example we called as weeds. Weeds also is a one one of the example for the up type it is. So these are the some significance can be possible whenever if you follow the minimum distance between the one field to the another field. So here again the minimum distance is dependent on the intended use. That's mean decreased isolation distance. Decreased isolation distance can be permitted with the following the situation, with the following the condition or circumstances. We can able to reduce the distance. For example, a presence of the physical barrier. Whenever you are doing the seed production with the help of the physical barrier, we can able to reduce the distance. So that is accepted. Then staggered flowering time. Whenever you are having the different planting time or flowering time, in that cases, decreased isolation distance can be recommendable to the particular person for the quality seed production. Then collection of the seed from the center of the block. Whenever you are collecting the seed from the center of the block, in these cases, right, the decreased isolation distance can be recommended to the given seed. Then similar crop type. Whenever you are taking the similar crop type like potato in the given region or maize in the given region, right, in that cases decreased isolation distance are highly recommended. That is the one we have. And increased isolation distance is necessary when we are having the following the condition. This means whenever you are having the larger number of the pollinators, in the given ecosystem, so pollen contamination is more in generally. That is the reason the more distance we need to maintain between the one field to the another field it is. Then whenever you are having the larger crop population, in that cases isolation distance should be more. Then whenever you are having the dissimilar crop type in the given ecosystem, we are having the dissimilar matlab brinjal, potato, maize, sugarcane. Whenever you are having the dissimilar type of the crop type, in that cases more isolation distance is required. See, these are the conditions we have under which increase the isolation distance and decrease the isolation distance are recommended to the particular person cities. Then, so some of the factors which affect the isolation, the distance, because one is the pollen behavior of the crop can determine the overall mechanism of the isolation distance. Then pollinating agent can affect the isolation distance, designated pest and disease in the given crop, then type of the seed production, right? We're having the seed production, some are the foundation seed production, some are the breeder seed, some are the nucleus seed production, then classes of the seed which is used for the seed production, then pollen trail and wide pollinators. So whenever we are having these factors, so sometimes these factors can affect the isolation distance in the given seed production technique. See, whenever if you follow this kind of the isolation distance we're having, so it helps in maintaining the physical and genetic purity. Maintaining the physical and genetic purity can be enhanceable in the given crop or seed can be possible. See, these are the few things we have in the case of the isolation distance. Maximum we can expect 10 marks in the given topic, not more than that. So as of now in the old UPSC paper, they asked 2 times about the questions on the isolation distance. In this case, the minimum distance is maintained between the one field to the another field, because our objective here is to minimize the contamination and maximize the homogeneity in the seed lot. So it is one of the method in the case of the genetic principle, in the case of the quality seed production. So we said in the previous class, within the quality seed production, we are having the 2 principles. One is the genetic principle, another is the agronomic principle. So in the genetic principle, we are having the various techniques. So among the various techniques, isolation distance is one of the technique. With the help of this technique, we can able to minimize the kind of the contamination in the given quality seed production. It's all today in the given topic. Thank you.